thank you all for having me here. So as mentioned, indeed, I'm a group leader since mid-2022, so it still feels very new to me. I'm in the lovely city of Leuven, where students pour wisdom and/or beer inside their heads, as you can see in the statue. We've got a very young team, very young group, so I can all just show them in one slide. 3 PhD students, a lab tech, and 1 postdoc that recently joined us. If you happen to be in Leuven next year, we're celebrating the 600th birthday of the university, so do visit us. We're in new buildings as well, which is really nice. As mentioned, so what we do is we're interested in variation. In part, it's somatic variation, so the mutations that accumulate in our cells throughout life. And we've got a particular knack for complex variation, which is kind of what landed me here in this session as well, obviously. Of course, I've looked at a lot of variants. We've done whole genome sequencing. I'll show you a bit of the work looking at cancer genomes, where we typically just chart all of the variation. We look at all of the SNVs, all of the indels, well, the ones we can see at least with short reads. But it's very dry. It gets dry very quickly. You see all of this variation, but you can't really figure out what it is doing without any other omics tied in. And so that's why we're trying to bridge a lot of the genotypes that we do capture, that we can see, onto phenotypes. And we use multi-omics technologies for that. Of course, we need to do a lot of technology development ourselves, and that is both in the wet and the dry lab. And I hope to talk to you about a bit of that in this talk. What has gotten us into all of this? Well, I used to work in the pan-cancer analysis of whole genomes consortium, which was an international consortium looking at 2,600 whole genome sequenced tumors, so both tumor and normal sample. And we looked at all kinds of variation in there, tried to reconstruct the somatic evolutionary histories of these tumors, etc. Of course, we landed ourselves in trouble very often by looking at complex variation. And I've just listed 3 of these ones. That took us years to put together software to tease apart what was really going on, working from short reads, which we could have actually solved really quickly in a matter of weeks if we had had long reads of these cases. First one is biallelic mutations, where the exact same position gets hit to the same type of mutation, to the same type of allele, or actually a different variant on both of the haplotypes. And if you can't phase your reads, that is very difficult to tease out if you've got a tumor sample that is not entirely pure either, which was the case in our samples. Another one is we actively went hunting in repetitive regions, tried to identify variants there, and we developed this whole thesaurus approach using software to put together reads aligning to homologous regions in the genome, and then see what fraction of those were mutated. Obviously, that allowed us to tease out the variation, but we couldn't really pinpoint it to any of those unique sites. And a last one that I want to highlight is looking at transposable elements. So these elements sometimes reactivate during tumor evolution, and they can jump around. And by doing that, they can cause additional damage to our genomes. So in this case, for instance, on the right, you can see a transposable element insertion. In this case, it's a line 1 insertion that has gone awry, and so it has resulted in a 20 megabase deletion that includes the CDKN2A tumor suppressor gene. And ultimately, the element ends up bridging these 2 chromosomes. So this is complex variation, somatic, actually, that we can start looking at with long reads as well, or that we hope to start looking at with long reads. So this is obvious. It's why I'm here. These kinds of cases would have been very, very easy to pick out. We would have just looked in our genome browser, seen the event, and actually said, oh, this is what's been going on. Long reads, we use both of the technologies. So we use Oxford nanopore sequencing. We use HiFi sequencing, both to call the variants, phase all the variants, do some de novo assembly when things get really tricky, but also to look at modifications. And I'll be talking to you a lot about that. We haven't done so much long-read transcriptomics work in our cases, but it's something we are inclined to do in the near future. All right, I've picked out one project that we've been involved in to kind of walk you through and show off some of the things that we do in the lab. And this is actually all about next-generation functional genomics. As we all know, functional genomics is hard. It's still very low throughput. You identify the variants, and then you go into the lab trying to figure out what they're doing, if anything. By using long reads and long read multi-omics specifically, as well as single-cell approaches, we hope to make this a bit more high throughput, actually a lot more high throughput. And the work I'll be talking to you about today is not anything somatic. We are looking at somatics in this project as well, but it's not the one I'll be presenting. It's Parkinson's disease, so neurodegenerative disease, very common. This is a project that started when I was a postdoc in the lab of Stijn Aerts in Leuven. It's actually headed by Thierry Voet, Stijn Aerts, and Christos Prokakis, who's in London at UCL. What we're trying to do here is actually figure out what's going on at the genome-wide association study loci that have been identified in this disease. So there's about 90 loci in our genome that have got SNPs in there that have been associated with the disease. And for most of these are tagging SNPs that have been identified. They associate with an increased disease risk, but we really don't know what the genes involved are in most of these cases. They're just tagging SNPs. They are most likely not the causal variants. Instead, what we believe is that there are more complex structural variants, repeat expansions, these kind of things that are messing up gene regulation at these loci. And so this project was specifically designed to try and figure out what is going on there. So what we have is a pretty large cohort. So this is about 200 donors that we've sequenced so far, control samples, healthy people across a wide, different range of ages, as well as people with Parkinson's disease. We've got brain samples, post-mortem tissue, and colon tissue as well, because this is believed to be involved in an early stage of the disease. We've done long-read, multi-omic whole genome sequencing on all of these patients. And from these same tissue bits, we've taken single cells, extracted nuclei, and done single-cell multi-omic sequencing. And this is ATAC sequencing, which will reveal the bits of open chromatin, and RNA sequencing. What do we hope to do with that? Well, as I said, we're trying to figure out what the variants are in these complex regions, these regions which have been associated with Parkinson's disease, find out all of the variants, and then add in these functional bits of information from the single-cell multi-omics, from the methylation, et cetera, as well. So what we have is, let me see if this pointer works. Yes, there we go. So what we do with the long reads, we reconstruct the two haplotypes. We found all of the variants, complex insertions, indels, SNPs, et cetera. We figure out what the methylation states are, which tells us something about the functionality of these alleles. And we tie in the single-cell multi-omic data, all of the RNA, all of the ATAC, which is something we can do then, analyzing that single-cell data based on this reconstructed diploid genome, allowing us to look at all of the variants that are not present in the reference genome. Haven't told you everything, so we're interested in multi-omics, and we push this as far as we can with these technologies. So what we're actually using for long-read, multi-omic long-read sequencing is this FiberSeq, which is an absolutely amazing technology developed by the lab of Andrew Stergakis in Washington. It was published in 2020. And what we do is we take cells, we take nuclei from our tissues, and we incubate those nuclei with an adenosine methyltransferase, bacterial enzyme that will put methyl groups on exposed adenosine bases in these genomes. We then extract high molecular weight DNA and sequence on our favorite long-read sequencing technologies. We get long reads out. We get whole, long haplotypes out. We get the endogenous CpG methylation, which already tells us something about the function. But we also get footprints. We get the open chromatin single molecule level, which is absolutely amazing to look at, and I hope to convince you later. This is not that straightforward, sadly. So if you do nanopore sequencing, this modification signal is a bit convoluted with the actual base calls, which can introduce some noise in the base calls and can introduce some artifactual mutations. And it's one of the reasons why we like PacBio HiFi sequencing, because there the signal is separated. So the base calling signal you get from the fluorescence, and the modification calls you get from polymerase kinetics. So this is more orthogonal, and it introduces less noise. But we get the genotypes, we get the methylation states, we get open chromatin states, and that gives us these amazing images of the chromatin in these cells. So what you see, for those of you who aren't used to looking at these kinds of pictures, I've got the reads aligned here on the two haplotypes. So every horizontal bar is a single read. These red bars is the endogenous CpG methylation, and these green stretches are stretches of methylated adenosine bases. So this is the stuff we introduced by labeling open chromatin. I've phased the reads into the two haplotypes. So you see haplotype 1, haplotype 2. And obviously, one of the first control regions that we look at is an imprinted gene. And so this is the gene house locus. So you can see the gene promoter end up here. And what you can nicely see is that there's a very big functional difference between the two haplotypes. So whereas haplotype 1 is hypermethylated, it's got close chromatin, so a lot of the non-green bases. You can see the chromatin is relatively open on the haplotype 2, and there's hypomethylation. So you instantly see functional differences between the two alleles. And this is really what we need to identify functional variants, and ideally complex variants. You can push this so much further still, because you can start looking at stochasticity, single molecule resolution stochasticity. And this is data from a cell line. You would think, oh, it's nice and homogeneous, it all behaves the same, but the opposite is true. This is a CTCF binding site, so a very typical clean example normally, where there's a binding site right at the middle. And this time I've grouped my reads by the functionality that we observed there. So in the top ones, this middle CTCF binding site is protected from adenosine methylation. CTCF is sitting there. It's nicely organizing the chromatin on either side. So you see accessible chromatin, then a very strongly positioned nucleosome, slightly less well-positioned nucleosome, and another one. And then there's two reads. Don't show any trace of having a CTCF molecule bound there. Just chromatin, one chromatin fiber, another chromatin fiber. So stochasticity, in this case, no CTCF. The bottom batch of reads, CTCF seems to have fallen off, but actually chromatin structure is pretty well maintained on either side. So this is telling us something functional as well, right? We could have a mutation at the CTCF binding site that makes it bind slightly less well, and then we'd see a shift in this distribution. And these are the kinds of things we hope to learn by doing this multi-omic long-read sequencing. Obviously, we've done a lot of toolkit building. We've developed pipelines because we need to deal with large amounts of data and haven't even talked about the single cell data yet. This is a lot, and we need these things to be scalable. So we've been building tools based on Aptainer, so containerizing tools. Put it all on GitHub. We use Nextflow and Docker to make everything scalable, neatly behaved, reproducible. I won't go into the details, but what we need to do in our case is take all of these whole genome data. We need to align them, get all the variants out, or in complex regions, actually push this into a de novo assembly, ideally local assembly in regions where we really need it. We can't work with alignments. We take all of the variants, and we push this to the single cell pipeline. Just to save costs, we actually pool multiple donors in a single lane and then need to demultiplex those donors afterwards. That's what we use the variants, the small variants for initially. And that will allow us to get those personal genomes out, take the single cell data for that donor out from the pool, and then map that data back to these personalized diploid genomes. What that allows us to do is start learning from these data, right? And this is actually a bit of work from Olga Sigalova in the ARTS lab, who's been taking those personal genomes, those reconstructed haplotypes that we get out from our donors. And by looking at the sequence, what she can do is actually train deep learning models, and this is based on convolutional neural networks, where we look at cell type specific enhancers. So an enhancer region that is just acting as an enhancer, say in some type of neurons or in glial cells, whatever. And this is what we use to train that model. And then we feed in the reference allele, we feed in the alternate allele of the enhancer that we found, and we see whether the model predicts any differences or any shifts in activity. And so we can very rapidly screen large numbers of genomes and try and figure out whether there's any changes predicted by the model in activity. And so, for instance, the reference allele, you can see a very low height of these letters, indicating that the reference allele is not predicted to be very active. The alt allele, however, in this case, and this is just a small snip, is showing higher activities, and we can then check that in the attack-seq data from all of these patients faced by their haplotypes. So patients which are homozygous for the reference allele, they don't show any attack-seq pileup. Patients homozygous for the alt allele, they show quite a bit of attack-seq signal. Patients that are heterozygous, they show intermediate signal. So we can validate this in the data instantly, these kinds of predictions. All nice. This is just a simple SNP example. Can we validate this in the long-read multi-omic data? Yes, of course we can. We can see these kinds of things appearing in there as well. Slightly different variant, but this is a case that we knew from beforehand. So again, I phased my reads into the two haplotypes. On top, you see the wild-type haplotype, and you can see that in the inset, so wild-type reads. And then mutant reads, which have a G at this site. Interestingly, this is a binding site for a transcription factor called GAP-BPA. where you can see if you have the wild type, you see more of these stretches of green, so accessible chromatin, open chromatin, most likely regulatory active. You see less methylation, endogenous CpG methylation on those sites, so we can actually see the wild type allele being more transcriptionally active than the alternate allele, instantly tying a genotype, in this case a pretty simple genotype, to a different function. And that's really what we want to get at. That's what we need to decipher these complex GWAS loci. Simple example, this works equally well, if not even better, with complex haplotypes. In this case, there's a repeat expansion at the top allele. You can see that there's about 1,600 base pairs of repeat insert here. You can instantly see the signal of hypermethylation on this allele. No way you could see this using short reads. Bisulfite reads, I wouldn't even start with. This ties a complex haplotype to a difference in function, and we can then check in the single-cell RNA-seq, do we indeed see a decreased expression of the involving gene. One of the downsides, I have to say, is that long-read sequencing, it still uses a lot of material, so we have to do these kinds of things in bulk, but we can then start developing deconvolution algorithms. So if there is, for instance, from the single-cell data, we know a certain cell type has a region that is accessible or it's hypomethylated compared to other cell types, we can then look back into our long-read multi-omic data and actually just fish out the reads that show that same methylation or open chromatin pattern. And that's kind of what I'm showing here. So this is the MBP gene, which we know is hypermethylated in neurons, and it's hypomethylated in oligodendrocytes. If we look in our brain multi-omic data, multi-omic long-read sequencing data, you see a subset of reads that is hypermethylated on both haplotypes, indicating that it's just a subset of cells that is hypermethylated at that site. The nice thing is this is hugely complementary, right? From the attack-seq, we just get tiny, tiny snapshots, short reads that show that this is open or this is methylated. From the long reads, we can fish that out, and we instantly get a 10 or multiple tens of kb snapshot of what the local chromatin state actually looked like in that cell type. So this is something we hope to expand on as well. One of the feelings I want to leave you with is that this is an absolute ton of data that we need to integrate, and we simply don't have the tools yet, which is why we're building a whole toolkit of methods on top of this. And so this is just a slide I've taken from one of my PhDs and the work packages that he set out. So we're building deep learning toolkits to go from sequence to these kind of single molecule chromatin patterns and actually predict these from the sequence alone, train on these kind of long-read multi-omic data sets, and that we can use to then plug in mutations, see what the effects would be of those types of sequences. We're working on models to actually classify these single molecules into this was an actively expressed gene or this was an actively expressed copy to this is silenced chromatin or whatever. And actually also applying computer vision approaches to basically take a look at all of these screenshots that I've just shown you from IGV and tell me what is going on. Is this actively expressed? Is this repressed? What is this doing? Just takes all of that information and plugs it into a single vector that we can easily interpret. With that, I think I'm very much running out of time. I'll just thank my lab, thank all of the labs I passed through over the years, highlight specifically our genomics course in Leuven as well, and thanks to our local funders, the FWO. We'll soon be getting a review there as well. So if you want to sequence with us in Leuven, talk to the people of the genomics core. We've got Wouter and Annelien sitting in the audience here. And thank you all for listening, and I'll be happy to take any questions.